Good afternoon. I'm David Patrick, and I'm a scientist and cardiologist in the research service at the Nashville Veterans Affairs Hospital and an assistant professor in the clinical pharmacology division at Vanderbilt University Medical Center. I'd like to highlight the important discoveries presented in our manuscript, isoleviglandins disrupt PU1-mediated C1q expression and promote autoimmunity and hypertension in systemic lupus erythematosus. The funding mechanisms that provided support for the study are listed here. First, I'd like to express my gratitude to the authors listed. These studies were a team effort and could not have been completed without this incredible group of scientists. The study focuses on the role of isoleviglandins or iso-LGs. Presented in this slide is the structure of the phospholipid of arachidonic acid. Upon oxidation, it undergoes rearrangement to form an iso-LG. Iso-LGs are highly reactive with the primary amine of lysine residues on proteins, forming a covalent bond, and thus a post-translationally modified protein. Importantly, there's a specific orally bioavailable scavenger of iso-LGs called 2-hydroxybenzylamine, or 2-HOBA. This prevents iso-LGs from binding to protein targets. Our study was based upon prior data that revealed the role of dendritic cell-specific iso-LGs, previously known as isoketols, as drivers of T-cell activation and organ injury in arterial hypertension. Among many important discoveries, this study revealed that the iso-LG scavenger 2-HOBA prevents dendritic cell iso-LG accumulation, T-cell activation, and hypertension. Additional important background information is presented here. We know from previous studies that lupus is associated with increased ROS production. We also know that complement deficiency is frequently observed in lupus and is a component of the SLIC criteria for the diagnosis of lupus. Importantly, more than 90% of patients with inherited C1q deficiency develop lupus. And finally, PU.1 is a transcription factor that's critical for the expression of the C1q gene cluster in dendritic cells. The purpose of this study was to determine the role of iso-LG formation in antigen-presenting cells in the pathogenesis of hypertension and systemic autoimmunity in lupus. The methods used to make these discoveries are presented here. We used human subjects, murine models of lupus, including the Black6, SLE123, and NZBWF1 mice, and additional in vitro methods. I want to specifically highlight that 2-HOBA was used to treat lupus-prone mice for studies of the effects of iso-LGs on hypertension and autoimmunity in lupus. So first, in human subjects with lupus, we observed a marked accumulation of iso-LG adducts in circulating antigen-presenting cells when compared to control subjects, as measured by flow cytometry and mass spectrometry. Additionally, using an ELISA to detect anti-iso-LG adduct antibodies, we found that the presence of these antibodies correlates with SLE disease activity index, suggesting that iso-LG adduct antibodies correlate with disease severity. Next, we observed that treatment of lupus-prone mice with 2-HOBA resulted in attenuation of lupus-associated hypertension as measured by radiotelemetry. We also found that 2-HOBA treatment attenuated renal injury, which may be observed on this Jones-Silver stain that highlights the basement membrane of the glomerulus and allows us to see immune complex deposition. Finally, we found a reduction in total circulating IgG and anti-double-stranded DNA antibody levels in lupus-prone mice treated with 2-HOBA. Mechanistically, based upon RNA sequencing from dendritic cells of lupus-prone mice, we discovered an important role of C1q and the C1q activating transcription factor PU.1. We found that iso-LG adduction of PU1 prevented its binding to the C1q locus, shown here in this shell shift assay. Single-cell sequencing performed on spleens allowed us to identify a cluster of C1q expressing monocyte-derived dendritic cells that expand in lupus and exhibit a pro-inflammatory gene signature that includes upregulation of the cytokine IL-1 beta. Importantly, 2-HOBA treatment attenuated expansion of these cells and their pro-inflammatory gene expression profile. The discoveries of the study are presented here in this model, whereby ROS leads to iso-LG formation. Iso-LG is then adducted to Pu.1 and additional cytoplasmic proteins. This leads to inhibition of C1q expression in monocyte-derived DCs and eventual DC activation and accumulation of apoptotic cell debris. Iso-LG adduction to these additional proteins leads to the presentation of these adducts to autoreactive T cells, leading to the production of autoantibodies and in-organ damage. Combined, these mechanisms contribute to clinical lupus. Novel concepts of the study are presented here. We found that iso-LGs accumulate in antigen-presenting cells in lupus and contribute to the production of novel anti-iso-LG adduct antibodies that correlate with lupus disease severity. Iso-LGs contribute to hypertension, renal injury, and systemic autoimmunity in lupus. Pu.1 is an iso-LG sensitive transcription factor, and iso-LG adduction of Pu.1 results in a reduction in C1q expression in dendritic cells. And finally, C1q expressing monocyte-derived dendritic cells expand in lupus and exhibit an inflammatory gene expression profile. And treatment with TUHOBA specifically attenuates expansion and inflammation of this unique cell type. The conclusion of the study is that iso-LGs contribute to systemic autoimmunity and hypertension in lupus by inhibiting Pu1 function in monocyte-derived DCs, thereby inducing a state of acquired C1q deficiency. Finally, the primary implication of the study is that patients with lupus who exhibit iso-LG accumulation or anti-iso-LG adduct antibody accumulation may benefit from treatment with iso-LG scavengers such as TUHOBA. Thank you for this opportunity to present our findings.